request first for our guests, our presenters, Dr. Yap, Sir Altomonte, Sir Gonzalez, and Dr. Rowsey to open their cameras. Thank you, everyone. Now we move on to our panel discussion to be moderated by our Professor Annalee Hadsall. For your questions, you can put them in the chat box, and our presenters will answer them accordingly. All right. So, Professor Annalee, you may take the floor.
Yes, we have one question here for Dr. Rowsey. Dr. Rowsey, okay, the question is there correlation between the distinctive features of the Ballotamus and the environmental conditions of Mount Compili?
Yeah, that's an excellent question. Thank you for asking. So much of the information we have about the ecology of Ballotamus is subject to some conjecture because we don't have a lot of information about how it behaves in life. Basically, the only information we know is that it visited traps that were baited with both fried coconut and live earthworms. However, based on what we know from other species and the traits that they share in common with these species, namely specifically shrew-like characteristics and the characteristics of tarsomes, the fact that it has this dark sort of reddish brown coloration both above and below suggests that it's likely active during the day. It has very dense, thick fur, which suggests that, or is typically an adaptation to the very mossy, high precipitation forests at these higher elevations. This long, pronounced snout with the large first molar and smaller second and third molars suggests that it might be an omnivore with a sort of insect and earthworm preference. So, but all of that is, again, just our best guess based on the little information we have from the 3 specimens that were collected.
Okay, then another question, same person from Aaron James Ortega. Do the distinctive features help Ballotamus compili thrive on its environment?
It's very likely. So especially if the species is an insectivore, or a vermivore as well, an animal that eats insects or earthworms. The earthworm and insect diversity of these montane regions is just incredible. In talking with Dr. Larry Haney, my former postdoc advisor, by far and away, like with mammals, the Philippines is an amazing hotspot of annelid or earthworm and leech diversity. So it's very likely that these adaptations allow it to specialize to some degree on some portion of the amazing insect and earthworm diversity that's found on Mount Compili. And who knows, maybe there's endemic communities of these earthworms in Mount Compili. My understanding is that no one's examined that yet.
Mm-hmm. Okay, thank you, Dr. Rowsey. And the next question is for all the speakers. Okay, question is, how can we sustain our collections in museums if we only receive little support from the university and the government? Okay, so this is a Miss Universe question. Anybody? Okay, I'll call Dr. Gonzalez first, then we'll have Dr. Yap, and last Dr. Rowsey. Okay, Dr. JC.
Thank you for that question. Am I loud and clear?
Yes.
Yes, there is support for collections from the university. It's a matter of actually requesting the support as well. Mahirap din kasi maghintay ng support. It's also good to probably go after the support itself. There's a lot of grants available. It's also grants available from the NCCA, I think. So it's just a matter of application for those particular grants. Also important to look into collaborations. There's a lot of international museums who would love to come to the Philippines, which is an amazing laboratory for evolution. And there's a lot of things we haven't studied. It's just the tip of the iceberg. And all of those collaborations can also bring in those much needed, not just expertise, but also the support, the new techniques, and of course, the grant that might be available through them. So I think, yeah, it's just a matter of expanding our collaboration as well as our reach towards awareness. Because the more awareness that we have, the more people would be interested in supporting museums. Next, Ma'am Sheikh.
Oh, yung pang universe question, Miss Universe. Because most of our taxonomic work are from our own pocket. Honestly speaking, yes. But yes, I agree with Dr. Gonzalez about collaborations. Actually, the one I have presented to you is actually a work of collaboration. Because here, taxonomists in Luzon and also taxonomists outside in the country that are really wanted to work with our what we have here in the Philippines need collaborations actually. Okay, so there are many international experts that are really looking at working in our country. Unfortunately, it's so that the one thing that also hinders them is the process on how to have their work here. We all know that getting permits and everything, all the documents to pursue all our taxonomic work, especially the field collection, is a very long process. I think to have a permit, it will take 2 years to accomplish that. And looking at if you have already a budget before you could make or you could go to the field, so the budget's already I think the funding is already done. It's finished already before your permit, before you have your permit. So I know there's some frustrations, but we have also students who are willing to help to continue our endeavors, our work in taxonomy, and to have more species to discover. So I agree with Dr. Gonzalez that collaboration is now the best way to work with this kind of research. So about
Thank you, Dr. Sheikh. Yes. Thank you, Dr. Sheikh. We'll see if that the same would work for Dr. Rowley. I think they have more financial support compared to Philippine taxonomies. Okay, Dr. Rowley. Dr. Rowley.
Yeah, I just want to echo what Dr. Gonzalez and Dr. Yap say, in that collaboration is absolutely key. The work to describe Ballotamus would not have been possible without collaboration, international collaboration with the Philippine Eagle Foundation and various folks in the Philippines. And so it definitely goes both ways. The work that this produced, the discovery and the description of this new species will produce specimens that are transferred to the Philippines and housed in the Philippine National Museum. So like this is an important generation of resources for the Philippines. The other thing that I wanted to sort of mention is that maybe this is just, me being overly optimistic, but I think globally, natural history museums are coming into what may end up being like a second golden age. I think of like the early era of like the late 1800s, early 1900s being like the first golden age for natural history collections. But I think we are entering this this era of discovering all of the breadth of different questions that can be asked and must be asked using vouchered natural history specimens. And there's a term for this, and it's called the extended specimen, where you can get from a single museum voucher, everything from parasite diversity to molecular data to protein information, dietary contents. And some of this information would be impossible to get without voucher specimens. So I'm going to post a link in the chat to a paper that describes this this sort of philosophy, and I think that that funding applications that propose to do field work with this in mind is sort of going to be the way that this that field work will start to head over the near to long term future.
Mr. John Altomonte is still with us? No more? Wala na siya, no? Mr. John Altomonte is still. Okay, I think he has left. Anyway, there's another question here from Mr. Reynante Bacalanco. Are there researches that indicate the presence of new species associated with climate change, or the other way around? Anybody can answer. Raise your hand and I'll give you 5 points for answering. Oh, I'm just joking. Anybody can answer. It's a free question.
I'll take a stab. So I think one area that deserves, that I'm not super familiar with and might be worth exploring is, at least within small mammals, there's a lot of, in the Philippines, there's a lot of historical data in certain regions at least. And so tracking shifts in distribution of different species along elevational gradients as we see global temperatures increase, I think would be a really interesting topic. So it's not so much a question of are there new species, but maybe where we see species is changing.
Okay, thank you.
I think there's a lot of things that I'll also echo what Dakota Rowsey was talking about in terms of elevational distributions. There's a lot of species, both amphibians and birds, which are montane species, and they're the most vulnerable. A lot of them are also restricted range species. Some of them may be endemic to a particular mountain. Some subspecies are very unique to certain mountain massifs, and then with the change in temperature may affect their distribution across that mountain or along with habitat change. I think a best example would be a species of frog called Ballotamus jesmosi, which was found in Mount Malinao. It's another place where Danny Belletti went, and we found that that particular frog was only found between 900 and 1100 meters. So it's just a narrow range, so that easily is restricted in terms, not just in one mountain, but also an elevational range. So that will be highly vulnerable to any change in temperature or habitat in terms of how wet those habitats are. A lot of species in the Philippines are also island endemics in itself. So island-based and mountain-based, any massive change, especially now with a lot of stronger super typhoons, can affect all those populations. And yeah, so again, the Philippines would be an ideal place to study any impacts of climate change across a lot of species, and because they're all integrated to each other, they're either pollinators or predators, or they're always integrated. It always affects another species. When you lose one, it's always a domino effect. So yeah, amazing places for study.
Okay, Dr. Yap. Okay, next question. This is for everybody. On your opinion, what are the significant areas of future-looking research in biodiversity research, considering your expertise, given that there will still be inherent challenges due to people's mobility? I'll repeat the question. What are the significant areas of future-looking research in biodiversity, considering your expertise, given that there will still be inherent challenges due to people's mobility? Yes, anybody can answer. This is worth 10 points.
Ma'am Annalee, can I go first on my group?
Giving a quiz. Yes.
on weevils, actually there are still more lot to study about this endemic genus, the Metapocertus. Actually, the most studied one that belongs to this on the same tribe is the Pacorhynchus. But for Metapocertus, we need to have phylogenetic analysis. We still have to study the mimicry. Then we have to have actually there's an initiative for the DNA barcoding of beetles around the world. So that's why there's collaborations, international collaborations for that. So if DNA, we just don't need, I think we don't need to go out. Most of the time, I think the initiative already was, they already have done the museum collections. They started with the museum collections. Then if ever we have some specimens locally, we could send to them, or we can have it here doing the lab work and send the sequences to include in the database. Okay. Also, there is the databasing. Then for the field work, actually, this is a good venue actually because I'm here in Luzon and the field work is in Mindanao, so I don't have to go there in Mindanao. The team of Dr. Amoroso do the field work there because they are there. So actually, I'm very thankful. That's why I'm very thankful with them for letting me in their team, even though I'm still here in Luzon. So what we did actually, I think Romeo is in the oceans, in the Zoom space. So during the descriptions of the new species, we are online. We do it online. So he sent me the pictures, or the what they do is then I told him, okay, take the pictures of these parts. Let's see what will be what it looks like. Then he will return back to me. So okay, so this could be a new species. Then I'll do the researching if it has the same characteristic with other species that I know, or the specimens that I have. So that's what the exchange of material, of the information what we are doing with Metapocertus.
Sir JC, would you like, or have you answered?
Okay. I agree. The Miss Universe question na naman ito. It's always, so I think it's rather than think of challenges, you always think of challenges as something that helps. It's much of a hindrance. It's actually an opportunity to change things. And for example, like sabi ni Ma'am Sheng is, if you don't have access to particular areas, and then it's an open door for collaboration with people who are in the local, in local universities or state universities who are there in those areas. And also helps build up training, not just for the faculty or the students of that university, but also for people involved in the field, especially if it's protected area. So it also opens up opportunity for capability building of people in the field, especially the ones working in protected areas, in the NR personnel. So I think it's, rather think of it as a challenge, let's think of something as an open door for something more integrative. I think Dr. Rowley actually mentioned about looking into molecular phylogenetics now is the age. It's kind of a new age for taxonomy. That we always think of taxonomy as these are little specimens in bottles. But now, because of molecular genetics and everything in systematics is quite new, we moved from Sanger sequencing to next gen sequencing, because it's a better method to look into short fragments. And so there's a lot of opportunities of expanding taxonomy as a science, using a lot more modern techniques, better microscopy, better looking into fluorescence in feathers. There's a lot of things that you can do and integrate. Again, if you don't have the expertise, I had to learn from scratch molecular systematics. That I can't teach old dogs new tricks. That's not true. So you can still learn a lot of new things and exciting things to do. But if you can't, then that's also an opportunity to collaborate with a geneticist or a molecular biologist. And if you're having difficulty understanding vectors and rasters, and work with a GIS expert. So it's all, again, rather than challenges, it has to be opportunities. Thank you.
Thanks,
So-
Dr. J.
Oh, I'm sorry.
Yeah,
Yeah,
Dr. Rowsey, your order.
My experience might be a little bit different as someone who's never conducted field work in the Philippines. But be that as it may, much of the work that produced the analysis of the new species, Ballotamus, was conducted during the height of the COVID pandemic. And so despite having, limited access to the physical specimens, much of the analytical stuff could be done from the comfort of my bedroom with my laptop. And that's one of the advantages, I would say, of some of the work that Museum Specimens offers, that as long as you have some initial access to specimens, which luckily I did prior to the pandemic, you can ask and answer any number of questions after collecting some data. So, given enough specimens, like Dr. Gonzalez was saying, you can go back and ask new questions with specimens that are 35, 50 years old. And so that's sort of the advantage, I would say, of natural history collections, is their function as libraries for folks who have limited ability to conduct field work. Certainly my work would be impossible without standing on the shoulders of giants like Danny and Dr. Larry Haney, who were involved with a lot of the field collecting.
Thank you. And then we have another question here. Well, I hope the three of you will not be, you're not involved here, no? The question is, how can you encourage personal collections of taxonomists to deposit their specimens in museums, or probably in herbaria? Personal collections. How can you encourage this personal collections of taxonomists? Have them deposit in museums, or if these are plants, in herbaria. So again, this is worth 20 points. I hope the two of you are not involved here. You don't have your personal collections. Okay, anybody can answer.
Okay. Would you like to try first?
Go ahead.
So I think it's in birds, there was that era of a lot of personal collections. A lot of the collections that I mentioned were mostly from museum expeditions, but earlier then there was a lot of kind of well-known people, or people with wealth, who had to show their wealth through menageries that they collect. So in the 1800s, like Lord Tweedale would have had Everett come to the Philippines and collect skins that would be part of their own personal collection. And a lot of them also had these amazing egg collections. But later on, all of these were then donated to the museums because they were having difficulty keeping them. I mean, keeping the collections themselves, unless you really, have an endowment of money to help keep it personally, then Yeah, it's very difficult and challenging. So a lot of the specimens were later donated to a lot of museums. I think that's the best way to how personal collections can be managed. I know that there's still a lot more in terms of, say, birds. There are still remaining personal collections. A lot of them would probably been, but again, that's one way to, of course, you have to ensure that your collections, your hard-earned collections, will be taken care of by that particular museum. So you need to look into that, the set of standards that they have in terms of upkeep, and make sure that they will be taken care of, especially if they have very rare specimens. I know if there's only one collection, which I probably deem personal, and they're now quite open as a museum themselves. So, thank you. Dakota?
Yeah, so at my institution, you know, we're in the middle of the Sonoran Desert, and there's a lot of reptile and amphibian diversity here, and there's sort of like a culture of folks who are just lay people who will drive down highways and find dead snakes along the side of the road. And this is a great opportunity to for them to contribute to the collections. And so some folks, like, they may not have like curated, you know, personal collections, but nevertheless, the goal is to indicate to these people that we will take care of the specimens that they collect, and that we will bring value to their material. And doing so like enthusiastically and by showing rather than telling is a great way to build allies with members of the public. And it has sort of a ripple effect, because, you know, they're oftentimes these members of the community, they talk to each other, and they go out in groups and try and find reptiles that are in, you know, new localities. And so by starting small and working with a couple people, like maybe a pest control manager who's interested in salvaging specimens, it has a sort of effect to, at least here in Arizona, to where there's a pretty nice culture of folks just from the public who donate specimens, at least with respect to reptiles and amphibians.
Thank you. And is Dr. Sheng with us? Dr. Sheng, she already left. There's a question for Dr. Sheng. Okay, so we'll reserve the question to Dr. Sheng. Anyway, this is something that I picked up from the chat box. These are reactions of some of our participants. Anyway, Dr. Sheng is not here to hear this. Anyway, from Romeo Patano, in Mindanao, the weevils are called, locals called weevils as kabayo-kabayo. It's a Filipino term, Dr. Rowsey. In English, it's just like horse or horse-like, because of the long rostrum or snout. Anyway, even if Dr. Sheng is not here, the question is for Dr. Rowsey. Dr. Rowsey, my question is for you. Okay, I think you have, if you cannot answer this, it could be a take-home exam, and you can contact your Filipino collaborator. There's something to do with the local name. Have you decided, or your group came up with a local name for your Ballotamus Compili?
Yeah, so in the manuscript, we actually, it's sort of a cop-out answer, but we actually suggest a common name, and we give two options, suggested options, either Mount Compili shrew mouse or Compili Ballotamus, which, you know, is just the Latin name but flipped backwards. So that would be my suggestion. Shrew mouse is a bit, you know, I've got my biases. It's not a shrew, it's definitely a mouse. So, but that's, you know, that's just my suggestion, or our suggestion.
Okay, thank you. Well, in addition to that, way back in 2016, we discovered this nice looking species that we named after the panelists. That's the handsome guy to your right, Dr. Rowsey, named after Dr. Juan Carlos Gonzalez, and that's a strong Gilodon, Juan Gonzalez. And people would call it purple. I mean, they would call it purple vine because the flowers are really purple. And sometimes they would call it the purple jade vine. When you talk of the jade vine, the color is really jade. So, the local name or the common name jade vine really is speaks of the species. In Masungi, people would be saying it's the purple jade vine. And until one day, they asked me if I can give a common name. There was no common name. So thinking it was, we named it after Dr. Juan Carlos Gonzalez. So it's now officially called, the local name is JC's Vine after his nickname, JC. Yes. Okay. So no more purple jade vine. Purple na, then you have a jade pa. Okay. Sir JC, would you add, you want to add something?
Actually, I think your question is referring to its Filipino name. So because the jade vine is called Tayabak, as you see on the 5 peso coin, which is the jade vine. And because also that the type locality of the original jade vine, Songgili the Makar Waters, is Matmakiling. So it's an amazing, it's a beautiful plant. And then thank you for naming the purple one after me. And I still have to see one in the wild. I still have to face JC Vine personally, face to face. So I'll probably have to visit Masungi or the ones in Diliman to see the vine, have a selfie with the JC Vine myself. But there's one interesting local name that may be useful for Ballotamus, is because you mentioned it's shrew and mouse, because often it's the local name Bubwit. Bubwit refers to a small mammal, which can either be a bubwit, which is the mouse, or bubwit, which is the shrew or the house shrew. So can be both.
Can you spell that out for me, please?
It's bubwit, the B-U-B-U-W-I. Please correct me if I'm pronouncing it properly. Annalee, tama ba bubwit?
Yes.
Okay.
Bubuwit, yeah.
But it's kind of spoken quickly, so it's pronounced fast. Bubwit.
Yeah. Bubwit.
Okay. So we have, yes, Dr.
I'm sorry. One more thing that I wanted to mention. I really like the local name for the weevils, caballo. I recognize that in Spanish the word caballo means horse, and so I saw that and I was like, oh yeah, I can see why it would be called horse-like. Actually, it's like 8:30 at night here. But I sent a message to my one of the other people I work with who studies weevils, and shared that bit of information with him. I thought it was super cool.
Yeah, okay. And then there's another question. Let's see it here. Okay. This is from Hazel Scott. We've heard about many new discoveries today, but what priority do researchers give into going back to existing collections for new insights?
So I would say that there's a high priority for that. In fact, many of the projects that I am currently involved with are doing exactly that. And I can give an example from some North American mammals. So it was recently found in the collection that I manage, our mammal collection at Arizona State, that there's this cute critter called an Ebert squirrel. They're a tree squirrel that is, found in sort of montane forests throughout Western United States. And here at ASU, we had about 300 of these squirrels that were collected in the 60s and 70s and had not been cataloged, were just like sitting in a cabinet for, you know, who knows how long. So it has been a great opportunity not only to train students to perform duties associated with managing natural history collections, digitizing specimens, etc. But it's also opened up questions with, you know, how can we use this treasure trove of data to answer questions that we couldn't 50 years ago. So we can take skin samples of these squirrels, get DNA from them, look at population genetics. And then if you combine that with modern samples, we can look at, we've got, you know, a 50-year time slice, essentially. So how have these specimens changed, or how's the species changed in terms of where it's found, what its genetic makeup is from the 70s to today? We can look at viruses just from the skin samples and numerous other questions. But personally, I think it's really important to get students involved with this so we can express, you know, this is, you could build an entire career out of this.
Sir Casey, your turn.
Yes, thank you. Yes, I agree. There's a lot of things you can work on with old collections. I mean, I don't think I sort of refer them as old. They're all collections. I mean, they're just a matter of historical data connected to them. Because in my case, although the question says not barcoding, but yeah, I work with historical DNA. I've actually extracted DNA from specimens that were collected by Wallace 150 years ago, and you still get amazing. They may be fragmented, but they are still workable. We can still get smaller fragments and concatenate them, and of course, still get amazing data. And then, especially with species that are hard to find. In my case, I had to get sequences from the Tikal Turictic hornbill, which is already extinct in the wild. So there's no way that you can get contemporary samples from any of them. And it's only with these very old collections that were collected by Robor 50 years ago that you can manage to get that information. In addition to those which are connected to the specimens themselves, that's what a study that was done in the Bishop Museum. I'm looking into hornbill specimens, and they actually were able to still get the Malophaga lice still attached to them, even though like 50, 60 years old. And that's what my student did, Ms. Fabregas, who went to the collections in Robor and found interesting Malophagan lice, 2 of which were new records for the Philippines. So yeah, there's a lot of things you can sift through, even just measuring the specimens themselves and looking into morphometric differences. And some of the collections may still harbor interesting new species or new taxa that may have been overlooked. That's what happened with Pinsker's hawk eagle. They were already pre-collected, and then people just notice, these plumage patterns are a bit, you know, interesting, and then realize that they've actually formed a new subspecies. So it's a matter of looking back. They're not just there to be kept in cabinets. They're there to be curated and studied, and of course they'll be useful for many generations ahead.
Okay, thank you. This is the second question of Hazel Scott. What are the trending insights that researchers have recently been looking for in old collections? This is aside from DNA barcoding.
So I think it really depends on the specialty of the person who is looking at things, which is like a terrible answer. But I've noticed, especially in the post-COVID-19 world, there's a huge emphasis on viral discovery, not only for being able to predict the next pandemic, but also just to understand how many viruses are found in species, in, you know, wildlife, and how many of them are unknown to science. Well, the answer to the latter question is almost all of them. there's recent work that I've started with some other folks here at ASU, where just looking at North American small mammals, we did just a brief, quick and dirty sequence workflow for circular DNA viruses, one just subset of viruses, and found just in a single rat, we found over 200 different species, we'll call them OTUs, operational taxonomic units, of viruses that had never before been sequenced. And we can tell that based on how different these viruses are from both each other and from known viruses. So you can imagine that's from a single rat. If you expand this across even one species, then you could get, potentially thousands of undescribed viral taxa, some of which may be pathogenic. So there's clearly lots of work to be done.
So JC, your turn.
Yeah, there's still a lot of things that can be studied in terms of genomics. I think that's what is the study we're being done with the CAPES program. They're going to metagenomics. So we need to look into how things are related between species, and how certain, it's not just looking into the specimen themselves, but the specimen's tissues, whether it's blood or even excrement. There's data in bat feces. It's what the study of CAPES is doing now with Project 4. So stay tuned for that. And this afternoon, that would be the next discussion on the CAPES program. So they'll be giving an update on that. So yeah, the same thing with all these other birds that it's not just the DNA of the birds themselves, but the DNA of all the other associated organisms that you may find in it, like the ectoparasites. There's a lot of these coevolution studies, whether it's a red queen hypothesis you want to test. It's amazing. There's still things we haven't done, whether those evolutionary relationships are more up close. And someone actually mentioned earlier about pollinators and how things can be associated with a particular specimen. Maybe those specimens may still have some pollen in the face or in the beak, or in the feathers, and you can still get those pollen and identify. It's not just barcoding, it's also just microscopy and looking into the specimens that you might find on those skins. So yeah, it's a lot of integrative studies that can be done.
Very much. I think, before we call on the next, the closing remarks, Sir JC, there's something here. I take a participant, somebody from the audience, Ariel Larona, was suggesting another common name for Strongylodon Juan Gonzalez, because you were saying jade vine, our Strongylodon macrobotrys is locally known as tayabak. That's the Filipino term, Dr. Rowsey. And since this is Strongylodon Juan Gonzalez, so Ariel is suggesting the first name of Dr. Gonzalez is Juan. So it's Juan Tayabak. That's nice, Sir JC. Anyway, we can build in a lot of common names. So it's Juan Tayabak now. Whenever you see this Juan Gonzalez entering Makiling, it will be called as Juan Tayabak, not JC's vine. Okay, thank you very much. And John Dexter Lumibao, I'm going to turn over the screen to you, John. Thank you both of you. Okay.
All right. Thank you very much to our speakers, our moderator, Professor Annalee Hadsall. And on behalf of the UPLB Museum of Natural History, I would like to thank everyone for, again, attending our morning series of webinars. And we hope to see you again later for our afternoon series of webinars.